Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Priyom, a postdoc from Duke University, and I will be moderating today with Dr. Yiqun Wang, a postdoc from UC San Diego. We're very excited to highlight the work of our amazing postdoctoral members. Today, Jennifer Watts from the National Children's Hospital and Dr. Bezia Lemma from Princeton University will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A, and please type your questions in the Zoom Q&A box. And with that, it is my great pleasure to introduce Dr. Jennifer Watts from the Nationwide Children's Hospital. Dr. Watts did her PhD at Michigan State University, where she studied the effect of Zika virus on embryogenesis. Prior to her research, there were very few studies that looked at the effects of the virus on pre-implantation development, and her work made significant progress on the topic by showing how different blastocyst lineages are susceptible to the infection. Her work has been covered by multiple news outlets, and she has an interview in development that you can check out to learn more about her work and her scientific journey. Dr. Watts is an active member of multiple scientific organizations, including the Alliance for Graduate Education and Professoriate, and has served as part of their steering committees along with organizing conferences for student success. It is a testament to her amazing service career that she has won multiple awards, including the Michigan State University Department Service Award, and as well as Outstanding Mentor and Teaching Awards. She also serves on the Early Career Editorial Review Board for Developmental Biology. Currently, she is pursuing her training in neurobiology, where she studies the role of GPI anchor biosynthesis in brain development, and we'll hear more about that work today. And with that, the floor is all yours, Jennifer.
Thank you so much for the kind introduction. That was amazing. I also want to thank the organizers for picking me to be a part of this amazing seminar. So without further ado, I will go ahead and get started. So there has been an increased discovery of congenital disorders of glycosylation-related genes over the last few decades. And this is mainly due to the accessibility of whole genome and exome sequencing in the clinic. Not only that, this also provides a need and tells us that there's a need to actually study these post-translational modifications to really understand the disease and hopefully develop therapeutics for it. So today I'll be focusing on GPI anchors, which is a specific post-translational modification, and variants in the genes associated with this biosynthesis causes many brain abnormalities as well as neurological defects, such as developmental delay and seizures, as well as structural brain anomalies. So, what are GPI anchors? So, glycosyl phosphatidylainositol anchor biosynthesis, as you can see, is a very involved process of a post translational modification. There are about 28 enzymes involved where there is a buildup of this anchor, then the proteins attached to this anchor, and then is translocated to the plasma membrane to act as an adhesion molecule, a receptor, or a membrane enzyme. And one of the first enzymes within this pathway is PIG-A. I also want to note that variants, there are about 24 variants that causes these GPI deficiencies in this pathway, and only 9 have really been studied. So there's a lot of area to actually study more deeply about these GPI anchors. So back to PIG-A. PIG-A is located on the X chromosome. And so males are most affected by the mutation or variant in this gene. And it causes quite a few of central nervous system symptoms, as I talked about before. One thing that is very important to note is that it does cause infant mortality, which is something that I'm really passionate about and really want to get to the bottom of. However, GPI anchor biosynthesis is very understudied throughout development. And so that's what I've been training on in the last several years on my uh, postdoc passion project with Pig A, and hope to expand that more in my own lab. So, my presentation is going to be in two parts. One, I will be talking about specific conditional knockout schemes to then study how GPI anchor or what the requirements of GPI anchors are within these different brain regions. I'll look at morphology and behavior as well as molecular analysis. The second part of my presentation today is going to talk about gene therapy aspects. And then I'll also be looking at behavior and cellular and molecular analysis after to see if we can recover some of these phenotypes that we see. Since pig A is the first enzyme in a pathway, this is a way for us and myself more specifically to study GPI anchor biosynthesis as a whole. So I mentioned before that pig A is located on the X chromosome, which makes the mutation or the variant X-linked. So I talked about how males are most affected in humans, and also human females are carriers, but they don't have many of those phenotypes that we see in male patients. And this is because most of the literature shows that there is a close to 90 to 100% skewing to the correct pig A. However, we see a little bit difference in our mouse model. And so we have almost like an intermediate phenotype where, due to X inactivation, the females have some retention of pig A. Rather than males have all depleted pig A with our Cree scheme. So to really show that, a few years ago, we ablated pig A in the full central nervous system using a nesting Cree mouse. And what you can see is that our males die very early throughout development, and our females also die pretty early, but this is around weaning time. We also see that we have motor coordination dysfunction, cerebellar hypoplasia, and hypomyelination. And this is all recapitulating many of the human phenotypes that we see in human pig patients. So now my question is, which brain tissues require pig A expression the most? Excuse me. So what I did was I knocked out pig A in the forebrain, and we saw some mild cell migration defects. We also knocked out pig A in white matter cells or oligodendrocytes, which are important for depositing myelin, and we see a reduction in the myelin. One of the major findings that we had was when we knocked out pig A in the cerebellum, we had postnatal lethality as we saw with the full brain knockout. So that was very interesting to us. We also saw disorganization of Purkinje cells and granule cells. We also see less of them. So what this shows us is that pig A is absolutely required in the cerebellum for normal development and especially development of the cerebellum. So that was very interesting for us. We wanted to also see if we can compare this to another enzyme within this pathway. So as I said, PIGA is in the first enzyme within that pathway to act. PGAP2 is one of the last enzymes to act within this pathway before the protein is translocated to the plasma membrane. What we saw was that there were no brain abnormalities in these animals. And so we already see a difference in the activity of enzymes within the same pathway. So that was very interesting for us. An exciting thing was that this paper was recently accepted by iScience. So we are happy to have this be available to everyone pretty soon. So I'm going to switch gears and reiterate that we have actually a pretty good model to study human pig A symptoms as it recapitulates many of the human symptoms that we see. So how can we rescue pig A loss effects? We collaborated with a lab that is at our institution at Nationwide Children's Hospital in the Center of Gene Therapy, who happened to already have an AAV9 for pig A. And we had our mouse model, so we just joined together to do injections to see if we can rescue many of the phenotypes, mainly the survival of these mice. So what I really want to show is the biodistribution of AAV9 using a GFP. We see that we do a single injection in the brain and the expression goes pretty much everywhere. So what we did was we used the AAV9 with a pig A packaged into it, and we did injections into the brain at P1. We looked at survival and behavior, and we collected samples at the humane endpoint or at 6 months. So I'm going to split this data into two parts. One, I'm going to talk about the females first. So we have our wild type in a gray line. We have our mosaic untreated in our red line. And similar to our study several years ago, we see that the animals do not survive much after weaning. However, when we inject these mice with our AAV9, both in lower and higher doses, we see an increase in survival up to 6 months. We also see this as a dose-dependent manner. Since we had such amazing results with our higher dose, we went on ahead and used the higher dose for males because, as I showed you before, males do not survive much long. So we wanted to use that higher dose. We see that while we see an increase in survival to the 6 month time point to about 30%, while not as high as our mosaic, we do see that increase, a significant increase in survival in our males, in our male mutants. We then wanted to study what's happening molecularly. So we wanted to see the expression of pig A as well as if we can rescue myelin. And so what I'm showing you here is the expression of pig A in the cerebellum. And as you can see, it's nicely aligned in our wild type mouse. But in our mosaic mouse, we see that the pig A expression is indistinguishable from the background. They then looked at fluoromyelin to look at myelin in the cerebellum. We have this nice striations. However, in our mosaic, this is depleted. When we injected these mice at P1 and collected around P22 and P28, we see that we have a recovery of pig A expression in the cerebellum in both our mosaic and hemizygous. We also see that we have a recovery of our myelin, which was really exciting to see. We then wanted to look at behavior. So we used a rotarod to look at motor coordination. Since our mutant mice do not survive to 2 months, we do not have that data. However, we wanted to compare wild type to our mutant treated. And what we see is that there is no difference between our wild type and our mosaic. We also see that similarly in our hemizygous males treated animals. So really the last part that I want to talk about today is to really look at what's happening downstream of pig A. As I said before, this anchor is attaching to proteins as a post-translational modification. So I wanted to see if there was a difference in abundance of GPI anchor proteins between mutants and treated animals. So we collected brain lysates. We use a selection tool called aflatoxin that specifically attaches to the GPI anchor and which will pull down only GPI anchored proteins. We performed mass spec. And on the side, we have a list of GPI anchor proteins that came out of our proteomic data. And if you see a more black square, that means that we have low expression. If you see a more green, white, or green expression, we have expression close to wild type or over wild type. And so in our first column in the yellow bar, we have our mosaic. And then in our green bar, we have our mosaic treated. And we see more green in that column. We then have hemizygous, which is more black and gray. And then we have our hemizygous treated, which is more white and green. So we are seeing an increase overall in GPI anchored proteins. And many of the GPI anchored proteins are involved in axonal growth, axons, injury, regeneration of this, and also synapse formation. So I want to end this talk with some take-home messages. We have our full CNS conditional knockout, but we really wanted to do a genetic dissection of this by looking at different regions. And we saw that PIG-A requirements vary in these brain regions, and more specifically, PIG-A is absolutely required in the cerebellum for development of the whole animal. Then we saw loss of PIG-A does not, PGAP2, excuse me, does not cause any brain malformations. Another take-home message is that gene therapy, replacement therapy rescues many of those neurological phenotypes that we had, as well as rescuing the neuronal GPI anchor protein expression. And so just to shout out some of the work that I've presented today, this first one is actually, like I said, was accepted by iScience, but if you want to look at the early version, it is available on Art bioRxiv. The second part that I talked about with the gene therapy is also on bioRxiv and currently under revision. And so this is really exciting that this data is already available to us to you to read even further. And yeah, we're excited to get this out into the public. So some future goals that we have for this is that we can create a mouse model known for human PIG-A variants. And so I actually attempted this with iGonad, and you can actually see that there's very severe effects, including craniofacial effects in this mouse, which we also see in humans. And so a variant model with these brain effects could serve as a better preclinical model than our conditional knockouts that we currently have. Like I said before, there are plenty of understudied GPI anchor biosynthesis enzymes. And we can ask the question, do these requirements differ between the distinct enzymes? And we already see that with our PIG-A and PGAP2 studies that that I just showed you today. And then we want to explore, which we actually already started these experiments, the transcriptomics of PIG-A mutants and rescue experiments. pretty much happening downstream of the GPI anchor proteins. And so, some questions that we have: what are the differences between wild type and mutants, male and female mutants, and mutants and treated? We also can delineate the biodistribution of the AAV9 as well in the different tissues in the brain. So, we're excited to see that data, and yeah, we're looking forward to it. So, without further ado, I really want to thank the members of the lab for their help and support. Former lab members Jesus Leal and Anthony Koulianos had helped me quite a bit on some of these studies. So, I really want to thank them. Gene therapy, and also our funding sources, especially CDG Care, who's helped a lot with the last part of my presentation. Um, I'm on the job market for assistant professor opportunities, so I have my email contact and LinkedIn if you want to contact me if you know of any opportunities. And now I'm open for questions.
Thank you, Jennifer, for a great talk. We already have a couple of questions, but I'll just get us started. So, looking back at your like lethality curves, do you think um, uh, the time point at which you see the male versus female dying, do you think they're like correlated to some like developmental processes? Which like marks this point, and then related to that, is there like an intervention timing in which your therapy would like be more successful and so
Right, right.
on?
So that's
And
those.
I'll just like to add that please add your questions to the Q&A box while Jennifer answers. Thank you.
Yeah, yeah. So those are good questions. So the first question, just to reiterate, you're asking, is there any kind of developmental process in why the males are dying more than the females? Um, I honestly think it's the genetics. It's that there's some retention of pig A in the females compared to males, and that's probably why the females are surviving a bit longer, because they do have some function. As we've seen in Western blots, which I didn't get to show today, um, rather than males have a pretty much a complete depletion of pig A, and so that's why they're dying so early. And so your second part of your question is talking about when is a great time to intervene. Um, we ask that question. We ask ourselves that question all the time. We are doing a postnatal, and we're seeing pretty great results. And that would be more comparable to doing a injection in a newborn in a human patient. But we have kind of thought about maybe some possibilities of doing some in utero injections, just to see if we see any difference. I think there has been a study, a clinical trial in humans where they did deliver genes in utero. I'm not sure where that's at right now, but it's definitely in development and something we can move forward with.
That sounds great. So we have a question from Justin on that they really enjoyed your talk, but they were curious about what kind of cell migration defects you saw in the pig A knockouts.
Yes. So you must be talking about the forebrain. So in the forebrain, when we knocked out pig A in our animals, we did an EDU experiment, and we saw that a EDU pulse chase experiment, and what we saw was that the cells didn't necessarily migrate all the way to the top cortical layers of the brain. And so that's kind of the mild defect that we saw. We also looked at other cortical layer, such as CTIP2, which is kind of these deep layers in the neurons. And we also looked at the upper layer of the neurons using CUX1, and we saw some differences in the expression. There was less of those upper layers. So we think that there might be a neural migration defect in those studies. So yeah.
Our next question is from the panelist chat. So they liked your talk and they were curious about how stable are these GPI linked proteins? Could the gene therapy treatment during development persist into an adult?
Yes. So how stable are they? Stable, they're not actually that so the GPI anchored proteins, they actually go through a second process where they get cleaved off into and from the plasma membrane. However, um, they're still attached to the GPI, and so they can be pretty persistent even if they're not attached to the plasma membrane. And then the second part of the question, could gene therapy treatment during development persist into an adult? Um, yeah, that's a really good question. We believe that it could be stable. We don't have we don't have enough information about it. However, AAV9 has been studied for 20 years, and it has done actually pretty amazing things in muscular dystrophy um, issues, and the patients are surviving for very long periods of time. So we're excited to see how this can progress even further.
Sounds good. Our next question is, what would a PIG-A gene replacement therapy look like in humans? At what level would the intervention be?
Yes, so that's a good question. The gene therapy, we've been kind of thinking about that, what that would look like in humans. I think this would be more since we did direct injection into the brain. So how would that look like? It would be more systemic expression in uh in those studies with humans. But it could very well look the packaging of the gene therapy could very much look pretty much the same. And at what levels would the intervention be? As we said, I think it depends on the patient since they do have a wide range of phenotypes. I think we want to have people who are very severe and possibly do that injection soon after diagnosis in these patients. So um, we are really learning a lot about gene therapy and AAV9s. This is our first foray into that world. So we're learning every day about how this could be translated into humans. But we're pretty excited and optimistic about it.
I guess I can ask another quick question. So,
Mm.
are you like, are there any other target enzymes that you're interested in, or which enzyme do you think is your
Mm.
like next?
Yes. So I have my favorites. Pig A and Pig F2 were really fun to work with just because of the different phenotypes that we have. But Pig W is really interesting because it's pretty in the middle of the pathway. It's not when it's attaching to proteins, but it is just as severe as Pig A, but further along in the pathway. So I'm pretty interested in figuring out what type of mechanism, what GPI anchors are affected with the loss of Pig W. Pig T is actually facilitating the attachment of the protein to the anchor. And so we kind of understand why that is causing very severe effects, but some of those in the middle, we're just kind of figuring out kind of the severity of those variants, and I really want to understand kind of the biology of it. So that is something that I definitely want to expand on. But yeah, thank you for asking.
That sounds great, and thank you so much for your talk.
Thanks.
I will let our next moderator, Dr. Yiqun Wan, introduce our next speaker.
Okay, thank you, Priyom, and thank you, Jennifer, for the great talk. It is my pleasure to now introduce our second speaker today, Dr. Bezia Lemma from Princeton University, where he works with Celeste Nelson and Andrei Koshmalu. Bezia earned his PhD in physics from Harvard University, There, he studied active matter and cytoskeletal organization with Dan Needleman and Zvonimir Jojic. Transitioning to Princeton, he has been an NSF postdoctoral research fellow in biology, and his research sits at the intersection of developmental biology and physics, and combines microscopy, computational modeling, and experimental approaches to understand how energy metabolism and mechanical forces can shape tissue morphogenesis, which brings a new perspective into the field. So Dr. Lemma's work has been published in leading journals, and he was selected as the Held Mango plenary session speaker at the 2023 SDB annual meeting. His work has also been recognized with several awards, including presentation awards at SDB meetings in 2023 and 2024. Beyond his work in lab, Bezia has also been active in science communication and open science outreach, including hosting the Lemma Journal Club, which is a podcast focused on recent papers in biophysics and developmental biology. He also created Lab on the Chip, which is a resource focused on accessible and low-cost scientific technologies. So with that, I'll turn it over to Dr. Bezia Lemma to present his work on mitochondrial ATP patterns and tissue fooding. Bezia, you can share your screen and turn on your camera now.
Share screen. Okay, and then, okay. Thank you for the extremely flattering introduction. My name is Bezia Lemma, and I'm going to show you my work on the patterning of mitochondrial ATP during tissue folding. Before we get into the experiments, I want to drag you into maybe an unusual perspective. I want you to think about energy itself as a pattern driver of development and morphogenesis. So for instance, consider this actomyosin. Actomyosin uses the chemical of energy of ATP to do work, which is a form of mechanical energy. And it releases some heat in doing so. And the interesting thing about energy is that in some deep sense, all these things, the chemical energy, the mechanical work, the heat, have a single common currency that they exchange between themselves. Energy is all tracked in the same units, joules. And so when we look for patterns of energy, very different things can be related in some unifying ways. And so if energy is patterned, you could imagine that these energy patterns might shape and be shaped by signaling patterns, or you could imagine that energy might pattern and be shaped by mechanical patterns. And so this is a kind of heady concept, and we'll get back to it at the end of the talk. This talk is really about trying to see if something like an energy pattern even exists. So if we take that actomyosin that we talked about, one thing that actomyosin can do is it can form a mesh at one side of the cell and then use ATP to contract, right? So the chemical energy of ATP is turned into the mechanical energy of work. And I think in the introduction you were promised some simulation. So here's your simulation. I have just a slab of, let's say, an epithelial sheet. And in this circle, I've programmed some actomyosin. And this mesh, I've just kind of cut away half of the slab so you can see inside the slab. And so this epithelial tissue is going to contract where the actomyosin is. And when these cells all contract together, they generate a small fold in the sheet. So here you can see this small fold that they generate as they contract. And it's this particular conversion of chemical energy into mechanical deformation that I'm going to look at to try to see if I can see a pattern of energy here. The system that we use to look for this pattern of chemical energy for apical constriction is the chicken lung. And we landed on the chicken lung, so I'll just say what this is over here. On the left is a embryonic chicken lung, HH25, about 124 hours. And in orange is the epithelium. It has a trachea, which then splits into these bronchi. And these are these very initial branches. And we can spin this around and see these branches. And we ended up going with the system because it's very simple. There's no differentiation going on here. Proliferation isn't necessary. It's just this actomyosin-driven apical constriction. And the lung is just a simple layer of cells. And so this seemed like the ideal situation in which there's not 100 different things going on, but we can just watch, hey, this actomyosin constricts, and we see a fold. And so the first thing that we do, this is going to be a lot at once, so I'm going to try to go real slow. The first thing that we do is we just stained this tissue for mitochondrial outer membrane. Or sorry, for this TOM20, which is a mitochondrial membrane import receptor subunit. And so this is a whole mount immunostain for this essentially recorder of mitochondrial density. And I have on the right here just a reminder of how this system works with the contraction. So you can see these branches. And so already you can see that there's a lot more mitochondria in the region of epithelium than in the mesenchyme. And why are we looking at mitochondria? Well, the mitochondria produce ATP, but they're also really large and really easy to stain, whereas staining ATP is not a great idea. So this seemed like a really easy first pass. And if we look at Z slices of this image, if we see a region that isn't branching and is never going to branch, and we compare that to a region that either is branching or is about to branch, we see a much higher mitochondrial density, and we also see this kind of like unique mitochondria clustering up on the apical side. We can also, just to make this a little bit more believable, we can also take these lungs and look at spatial expression levels. So here is, some spatial RNA-seq, and I've kind of, to give you some bearing on where you are, I've put the mesenchyme in blue as this col1a, and I've used EpCAM to label the epithelium again in orange. And if we take all the mitochondrial genes and sum them together, we can see that these mitochondrial genes are being expressed significantly, I shouldn't say significantly, are being expressed here at these branch points. So there's like this surprisingly strong pattern of mitochondria in the early development of this organ. And there's a lot of directions that one could go from here, because I think that this result in itself is shocking. If I had known more about biology when I started this project, I would have not started this project, because I think one would have obviously said that this is not going to happen. So the fact that it does happen is super interesting. But I want to look at these systems from this perspective of chemical energy patterning. And so we're going to try to dig down further into this mitochondrial ATP production. The way we do this is we can dissect out a lung, and we can inject some dye, or we can also electroporate in genetic reporters. We make a little hole at the bottom of the lung so that when we inject in the dye, it doesn't inflate like a balloon. And we use this dye called ATP Red 1, which when I gave this talk in Chicago in 2023 was a brand new dye, and now it's been a while. So it's very well characterized. It doesn't fluoresce when it's attaching to ADP or GTP. It fluoresces specifically when attached to ATP. And we can wait for it to get into the epithelial cells, and then we can watch the mitochondria themselves wiggle around, because this dye really glues on to the mitochondria, because it's a slightly polar dye. And so that's why I call this mitochondrial ATPs, because we're really just reporting the ATP surrounding the mitochondria. But we have this intensity that's proportional to how much ATP is there. And so we can then get images like this. They're a little bit tricky to interpret because we are imaging live tissue now that's 200 microns thick. So a lot of what is bright and what is dark has to do with how much the tissue is occluding or how much other tissue is occluding the tissue that we're interested in. And also this mesenchyme on the outside is getting exposed to a lot of dye that just didn't get washed out. So to correct for all this, the way we think about this data is we compare a bit of the dorsal epithelium, that's the top bit, like dorsal, like a dolphin, and we have this ventral epithelium, and we say this dorsal and this ventral epithelium probably got exposed to similar amounts of dye. They probably have a similar amount of tissue occluding them. And so we report the ratio of the dorsal epithelium to the ventral epithelium. We're only really interested in variations along the dorsal side, which is where the branching is happening. And so we use the ventral side to normalize. And what we see after we do all this is that regions that are not branching and aren't going to branch have significantly less ATP red 1 than regions that are branching or are going to branch. And these are live movies, right? And so having these live movies and having this result that the ATP is increased in places that are branch or are going to branch, we wondered, you could imagine two ways that this goes. Either the apical constriction starts, there's some decrease in ATP, and the mitochondria ramp up and provide that ATP, and then you get your apical constriction. Or it could be that the mitochondria ramp up first, and then the constriction starts to happen. And either one is pretty interesting. No dog in this fight, although now I do because I say, if we measure the ATP red 1 intensity, we see that the ATP red 1 intensity, which is this red line, increases before the bending begins. So this black line shows the branch deflection. And so we have a couple hours of ATP red 1 intensity increase before the branching occurs. And so we say from this that the mitochondrial ATP production precedes the apical constriction event and then continues to increase during folding. So we then asked, if it precedes folding, are these mitochondria necessary for this folding? And we can use oligomycin, which inhibits ATP synthase, so it stops mitochondria from building ATP. And in the control, you can see that we have our 2 branches, and we wait 24 hours, and now we have a bunch of branches. When we expose the lung to oligomycin, we have our 2 branches, and we wait a bunch of hours, and we still just have 2 branches. And if we do some immunostaining, we see that the mitochondria are still there, but they've gotten kind of washed out in their signal. And the phosphorylated myosin light chain is no longer present on the apical surface of this branching region. So it appears to be necessary, and I actually thought this was a bizarre result, because it doesn't require that much energy to create a branch, that much mechanical energy to create a branch. And I thought that glycolysis would be able to deal with the slack, especially this early in development. And so this made me wonder just how much chemical energy was required to make this fold. And the way we got about measuring that is we know that mitochondria create about 5 ATP for each oxygen that they consume. And so we did oxygen consumption rate measurements using seahorse. We divided the lung into a proximal, a medial, and a distal region so that only the medial region is branching. And we pooled these together into this proximal, medial, and distal pools, and then we measured the oxygen consumption rate for each one of these pools. And again, the oxygen consumption rate is directly proportional to ATP concentration, and from that you can calculate, how much chemical energy per second, what is the power of each regions of these tissues. The proximal and the distal tissues are burning through the mitochondria are producing about 1 microjoule per second of chemical energy in the ATP. The medial tissue is about double that. It's producing about 2 microjoules per second. A little bit more, I'm rounding. So it is really producing the mitochondria in the medial region are really producing a lot more energy. And so we wanted to know how much of this is really related to the actomyosin-driven apical constriction. And we went in with another inhibitor, a myosin 2 inhibitor, blebbistatin. Blebbistatin, again, if you have your control, you get new branches. If you put in blebbistatin, you still get growth of the lung, but you don't get new branches. And what we saw from this, right, we're now inhibiting this actomyosin-driven apical constriction, is that there's a little bit of a hit to the proximal and the distal mitochondrial ATP production, but there's a huge hit to the medial mitochondrial ATP production. So we inhibit the actomyosin, and all of a sudden the mitochondria are producing half as much energy. So really, that 1 microjoule per second chunk here seems to be dedicated to this one folding kind of program. And so there appears to be patterns of chemical energy that exist, and they predict tissue folding. And so this was really exciting. I talked to you about the mitochondrial patterning itself, and then we measured mitochondrial membrane potential, which I didn't show here, and we measured mitochondrial ATP production, and we even did very slightly spatial measurements of the ATP phosphorylation rate by the mitochondria and also by glycolysis, although those experiments are a little bit iffier. What I want to talk to you about now is whether or not this is like broadly true, or is this like we did it in this one situation and it came out the way we wanted it to in one situation. So the quick answer is no. Here's green mitochondria for a fly, and in purple is myosin. The slow answer is right, like this mitochondrial enrichment pattern is conserved. Here in the fly during the formation of the ventral furrow, we see both live and through staining this enrichment of mitochondria along the furrow. We can see the same thing in the mouse eye, this enrichment of mitochondria. We can see the enrichment of ATP along the mitochondria in the mouse eye. And we can see the same thing in the chicken neural tube closure. Not the full closure, but the bending towards the closure. And again, we can see in purple the mitochondrial density is increased in this tissue, and the ATP along the mitochondria is increased in this tissue. We can just take these patterns and put them on to a similar geometry. So how I did with the actomyosin, that black patch, now we're just going to take this ATP red pattern and put it onto this geometry and say the more ATP you're expressing, the more you're contracting. That's all the simulation is. And we get, these surprisingly good recapitulations of the geometric changes that occur. So this is, getting close to folding. But I want to talk a little bit more about this eye picture, because this, I think, was one of the funnest moments that I've had in science in my career, which is I was asking people, I need an actomyosin-driven apical constriction event where there's not a bunch of other things going on. I don't want differentiation and proliferation. And I need it to be in the mouse because people really care about the mouse. And so eventually someone said, oh, the nasal placode does this. You could look at the nasal placode. And so I went and I stained a mouse head looking for the nasal placode. And every time you kind of look at a new system under a microscope, you don't know what you're looking at. And I saw this, and I was like, oh, is that a nasal placode? And someone was like, no, that's an eye. I'm like, oh, but it has the thing. And what did I mean by the thing? Well, it's not just that the mitochondria are enriched in the tissue, right? It is enriched in the tissue, but it's also like this very clear motif. The mitochondria are enriched in the tissue, and they all go to the apical side. And that's the thing that your eye picks up on when you're seeing this in microscopy. The nasal placode, by the way, also does this. It's just much less impressive. And so if you take any one of these systems that I showed you, there's always more mitochondria on the apical side. And sometimes there's a little basal population, and sometimes there's not, depending on the system. But I was trying to think about why this is what the motif looks like. And so you could imagine here in green is actomyosin, and here in purple is mitochondria. And here I'm going to have a little simulation of here are the mitochondria. The mitochondria take ADP and they turn it into ATP, and the actomyosin takes ATP and it turns it into ADP. And if the actomyosin is right here and the mitochondria are right here, you get a curve of ATP to ADP ratio that looks like this. And actually we can measure this inside the chicken lung using this Percival HR genetic sensor. But the thing that I think is interesting is what if the mitochondria wasn't there? And so we can imagine how well would the actomyosin work if the mitochondria moved away from the actomyosin. And so we can move this block of mitochondria away so that the amount of ATP that the actomyosin have is less. And we can see that according to Michaelis-Menten kinetics, the apical hydrolysis rate, the hydrolysis rate of the actomyosin is going to dip by a little bit less than 10%. And so there might be this energetic utility in putting your energy provider right next to your energy user. And that there's this, things will run faster if the ATP is more available. And so, okay, here's my one equation for the slide, or for the talk. Don't kill me, I'm going to try to go through this. I'd like to propose a length scale above which ATP phosphorylation is going to become spatially patterned, right? Glycolysis and mitochondria are going to become spatially patterned. When the length of your cell, and these are really long cells, these epithelial cells, is greater than some number set by the diffusion multiplied by how much time it takes to munch through the ATP pool, then the system might start putting the mitochondria right next to the region that's munching through the ATP pool. And if you look at other people's data, I think you can see this in a lot of different papers where there's a large population of mitochondria on one side of the cell, right next to a very energetically intensive process. And in our systems, just to put some numbers on this, the diffusion coefficient is about 100 microns squared per second. The hydrolysis occurs so quickly that it takes about 3 seconds for the actomyosin to go through kind of the 7 millimolar ATP pool of the cell. And so the length that we get out of that is 17 microns. And so when a cell gets above 20 microns, you might start to see it organize and spatially pattern its organelles to deal with this. That's my hypothesis that I'm just trying to throw out there to people to see what they think. And so, we see these spatial bioenergetic motifs. They're quite interesting. I think that there's a lot of them, not just to do with actomyosin-driven apical constriction. This is an example. We see it in other creatures, so it seems to be conserved across different systems. And kind of coming back to this eye, which really captivated me. The day before you have mitochondria enriched up here, and nothing going on here. The day of, the mitochondria are enriched here, and they kind of fade away from here. And so you could really imagine, these complicated ways in which signaling pattern energy. I think that's pretty obvious. But maybe what's less obvious is that energy can pattern signaling, right? That it can make certain signaling processes go faster as energy becomes more available. I think that this has been done a lot for how mechanics and genetics can intercede. And so maybe an easy way to see this is like, if you have some mechanical system that uses up energy to make a dense mesh, once the mesh gets dense, ATP can't diffuse in there anymore. So the system stops getting so dense, and you can have this kind of feedback between your mechanics and your structure and your forces and how much energy can cause further contraction. And so besides things like, a gene causes something to happen, which changes the mechanics, which changes the energy, there might also be these pathways in which like a supply of energy causes, some metabolite signaling, which then causes some mechanics. And so there might be these patterns going on in these systems that we're currently blind to that we'll be able to see in this manner. And so thanks to the Nelson and Koshmerl labs, thanks to everybody who helped me with this program. These are my chickens who inspired me to go work with Celeste Nelson. And this is my daughter who brought this work to a crashing halt about 2 years ago. And these people, particularly Dr. Megan Rothstein, I'm really grateful to for carrying the torch and getting this paper developed enough to publish while I was out on paternity leave. And thank you for your time.
Thank you, Bezia. That was a great talk. Very intriguing.
Leave it here.
I have a question. So you proposed the model at the end to suggest that there might be this optimal spatial distribution between the sites that is consuming energy and where the sites of energy production, mitochondria, is. I find that very interesting, but I also wonder whether you have any speculation in terms of molecular mechanism wise, how are mitochondria put at this optimal place? And that process probably is also consuming energy, right? Is that also considered in the model?
Yeah, so I think that this is like the very clear next, or I think that this is probably the most exciting question of where you could go with this immediately. And so I'll say kind of two things. One, for how the mitochondria get to where they would be most optimal. I don't think that this is conserved between species. You can see kind of in the shadows, there's this paper that came out last year showing that while the mitochondria, while Drosophila are forming their ventral fold, they do so with such like violence that the nuclei shove to one side. And you can see in the shadows of their videos, they don't label mitochondria, but you can see it anyways because mitochondria are so dense. You can see the mitochondria get shoved to the other side when that happens. the Drosophila in general, right? Like things are happening on the time scale of minutes, right? Tens of minutes. In the chick and the mouse, things are happening on the time scale of tens of hours, right? And so I don't think the process by which the mitochondria get moved to a side is going to be conserved. My wild speculation for how it works in the chick lung is that there's a miro system which connects miro to mitochondria in ATP rich areas that moves the mitochondria to ATP poor areas. That's wild speculation. I have no evidence of this. But that the mitochondrial patterns are not exactly the same from organ to organ or from species to species. Sometimes we have this basal population, sometimes we don't. So I think that there's a general underlying principle that says it's optimal to look in, to get this pattern, but there are many ways in which you can get this pattern. Yeah, and then similarly for the energy to set up the pattern, right, depending on the schema, depends on how much the energy considerations are going to play a role.
Very interesting. Can't wait for you to get into these questions. Another question from our Q&A from Natasha Shiloh. She wants to know that when she thinks about apical constrictions, she thinks of things getting denser. Do you think mitochondria also have similar roles for tissues which are expanding, kind of like a bloom? Right now, she's thinking of lumen formation in early human embryo, and it starts as a rosette that expands until a lumen fold.
Yeah, yeah. So here I'll say I think about it a lot and I have no speculation. So I'll just say some things. One is, particularly for Natasha, we've tried to look at this in anoles, and actually I've tried to look at this in one chameleon lung once. And the system is complicated. There's patterns that exist, but it's not this pattern, it's doing something else. Some other notes is a system that I've been really looking for is a basal wedging system, where instead of having apical constriction, you have on the other side basal wedging. It's, two sides of the same coin, but you would expect to see mitochondria on the other side if this like underlying motif was true, if like this spatial bioenergetic argument was true. And I haven't found a clean system to do that in, although I have plenty of candidates. And yeah, I mean, this is about as good as I can do for like actual like speculation about expanding and things like this. I just I get stuck in systems that have multiple things going on at once. So there are systems that are just doing proliferation or just doing differentiation or just doing something like this fourth generation. But once you start to layer those on top of each other, you have to have a really clear idea of how any individual thing works before you can layer them on top of each other. And so a lot of systems I kind of back away from.
Next, we have some more questions from our panelists. Billy Swala wants to ask, also related to the spatial patterning between the energy consumption and energy source. Is there any way to move the mitochondria away from the apical side and see if the tissue still constricts?
Yeah, so this is why I hope that it's the Miro one. We have an optogenetic Miro that we can get into this system, so we'll see if that works. And we'll see if we can move the mitochondria in that manner and find out about like sufficiency, right? We have some other schemes by which we will try to do this if that doesn't work. We also have this way to optogenetically induce apical constriction, and we'll see if we can see mitochondria move to the apical side. They're supposed to go there first, but we'll see what happens. But I think that these experiments are a long way off, that this is not going to come out 6 months from now. This is going to come out years from now.
Okay, so one last question from Jennifer Watts, our last speaker. She said, could increasing ATP or mitochondrial production get more branching? Can you increase the mitochondria and ATP production in the system and see what happens?
Yeah, so you could, right? You can put in a drug like FCCP and just like supercharge all the mitochondria. I'm always very whiny as an experimentalist. Everyone wants me to do these drug experiments, and I'm scared of the drug experiments because you change so much when you put in a drug. that I have difficulty thinking about what I've done.
Mhm.
I've never done it. I don't know what would happen. I really don't know what would happen. So it's an interesting idea and also great talk.
All right, I think that's all the time we have today. So thank you again, Jennifer and Bezia, for your excellent talks and for sharing your works with us. This seminar has been recorded and will be available on the SDB website next week. And today is the last Ethel Browne Harvey postdoctoral seminar in this season. We want to take this opportunity to thank all of the speakers who presented this season. Applicants for the 2026 to 2027 season will open in June. So thank you all for coming today, and we'll see you again in the fall.